Hi, come with me while I demonstrate one chemical method and one physical method of controlling microbial growth. The work area has been prepared using aseptic technique. With the sterile work area prepared, the materials are ready for use. There are TSA and EMB agar plates. Next are the sterile test tubes used to collect and hold a previously collected 5 millimeter sample of 21-day sauerkraut brine, which serves as the untreated control for this experiment. The remaining 21-day sauerkraut brine is contained in the original fermentation jar. Also included are 70% isopropyl rubbing alcohol for the chemical treatment, sterile 1 milliliter pipettes that were available if needed, and tape that will be used to seal the agar plates after streak plating. A measuring cup will be used to accurately measure and divide the remaining sauerkraut brine into 2 equal treatment samples. And finally, a thermometer will be used to verify that the hot water bath reaches 100 degrees Celsius and to monitor the temperature of the heat-treated brine as it cools back to approximately room temperature before streak plating. Before preparing the brine samples, the measuring cup was sterilized. Water was heated to boiling, and the measuring cup insert was placed into the boiling water for 5 minutes. After sterilization, the measuring cup insert was removed and allowed to cool before being placed back into the measuring cup sleeve. The boiling water temperature was checked using a thermometer to confirm that it had reached 100 degrees Celsius. The sterilized measuring cup insert was placed back into the measuring cup sleeve. The 21-day sauerkraut brine was then thoroughly mixed to ensure an even distribution of microorganisms throughout the sample, and the brine was transferred into the sterilized measuring cup, and the total volume was confirmed to be 118 milliliters. The brine was then divided equally into 2 portions by transferring half of the sample, approximately 59 milliliters, back into the original fermentation jar. This created 2 equal treatment samples, with one portion designated for the chemical treatment and the other portion designated for the physical treatment. The lid was replaced back on the jar containing the physical treatment sample, and the jar was placed into the 100 degrees Celsius boiling water bath. The timer was set for 10 minutes, and the jar was gently swirled throughout the heating period to maintain even mixing and heat distribution throughout the sample. After the 10-minute heating period was complete, the jar of brine was removed from the hot water bath and placed on a clean towel to cool back down to approximate room temperature. While the physical treatment sample was set aside to cool back down, the chemical treatment sample was created by combining 59 milliliters of 70% isopropyl rubbing alcohol with the 59 milliliters of sauerkraut brine remaining in the measuring cup to create a 1-to-1 mixture. The sample was mixed thoroughly to ensure even distribution of the chemical agent throughout the solution and was allowed to rest for 10 minutes. Following treatment, the chemical treated sample was streaked using a sterile cotton swab to transfer the sample into Section 1 of the TSA and EMB agar plates. An inoculating loop was then sterilized in the flame of a tea light candle for 10 seconds before streaking from Section 1 into Section 2 of the TSA plate. The inoculating loop was sterilized again for 10 seconds before streaking from Section 2 into Section 3 of the TSA plate. The inoculating loop was sterilized once more before repeating the same streaking and sterilization process on the EMB agar plate. After streaking was completed, the lids of the agar plates were secured using tape to keep the plates closed during incubation. The plates were then placed in Ziploc baggies, labeled, and left to incubate for 24 and 48 hours. Here it is shown that the agar plates are correctly labeled chemically treated brine. After completing the chemical treatment plates, the physical treatment sample was ready to be checked. The sample was gently swirled to ensure the brine remained evenly mixed, and the temperature was checked using a thermometer to confirm that it had been returned to the approximate room temperature. A sterile cotton swab was then used to transfer the physical treated brine into section 1 of both the TSA and EMB agar plates. The inoculating loop was sterilized for 10 seconds between streaking sections, and the streak plate process was completed to separate microbial growth across the agar surface. After streaking was completed, the lids of the agar plates were secured using tape to keep the plates closed during incubation. The plates were labeled correctly and placed in Ziploc bags for incubation. And that is it for my demonstration of one chemical method and one physical method of controlling microbial growth. The agars will be observed in 24 and 48 hours. The control brine was previously streaked on agar plates, and these are the results after 48 hours. Bye!